Hi class, so my name's Aiden McParland. I'm a senior here at UNE and I'm doing this neurobiology class as part of a 4+1 master's program. So I'm in my senior year, as I said, and next year I'll complete my master's, and I have a couple of extra assignments I have to do for the course, and this is one of them. So this is going to be a video sort of briefly summarizing these 4 papers that we're going to be presenting on or hearing presentations on this Tuesday, in hopes that this stimulates your interest maybe to read them a little bit, or at least to give you a little bit of a way to start or a place to start into how to dig into these papers, because they can be pretty complex, and some of the genetics is definitely pretty deep. And so, yeah, Dr. Gander asked if I'd give a quick overview of the papers. And as you know, in class we've been covering ion channels and the importance of these channels regarding the neuron's ability to signal and to elicit a response, whether it's to coordinate movement, or to devise a thought, or whether it's to avoid a noxious stimulus. So this set of 4 papers we've been assigned to read focuses on the discovery and understanding of the incredibly important potassium channel. So really, before the first paper that was published in this group, the Tenounier et al. Paper, we had yet to identify the gene responsible for encoding potassium channels, and we knew very little to nothing about their biochemical structures. So the way in which the scientists first knew where to look for this gene was that they observed a clear malfunction in the nervous system of fruit flies that had been anesthetized, because they exhibited this sort of shaky leg while the rest of the fruit fly was out cold. And they called these flies shaker mutants for obvious reasons. And now, after the famous fly lab in California called the Jan Lab hypothesized that it may be a result of a faulty potassium channel, the Tenounier team sought out to discover what was causing this phenotype, with a hypothesis that it may be a potassium channel defect. After using electrophysiology, in which we studied patch clamp in class, and some gene dosage analysis, which is really cool actually, and they insert wild type or healthy genes into these mutant flies in hopes that they can regain the normal phenotype of these action potentials, the researchers determined that they had in fact identified a defect in the gene encoding a component of the potassium channel. And so they then went on to understand where on the X chromosome this gene was located. And now that was kind of the summary of the first paper, the Tenounier paper. The second paper I read in this sequence was the Temple paper. And this group was interested in taking a genetic approach, which all these papers really look at this in a genetic way, to understand the biochemical structure of shaker channels. And they did so by making complementary DNA clones to different regions of the shaker gene, and they genetically sequenced these regions. And after doing this, the researchers were able to look at amino acid sequences coded by the complementary DNA, which allows researchers to make predictions of the structure of this protein. And the way this is done is that certain amino acids are hydrophobic, and so they don't like water, and they would fit in nicely to the cell membrane, whereas other amino acids are hydrophilic. And by determining regions where hydrophobic amino acids are coded for in simultaneous, or there's several sequences of hydrophobic amino acids, the researchers can kind of predict how many regions of this protein would likely be crossing the membrane, or how many transmembrane domains this protein has. And so yeah, if a sequence is part of the cell membrane, has a lot of hydrophobic parts, then it's a transmembrane domain, and like I said, they can determine kind of the overall structure of this protein. And the researchers in figure 6 of this Temple paper present an idea of what they think the shaker channel could look like. So check it out, it's pretty interesting. The other 1987 paper, so these 2 papers here were right after each other, was the Papazian paper. And this uses, again, a genetic approach to tackle exactly which channel the shaker gene is affecting, and to understand a little bit more about where this gene is located on the X chromosome. So they did some complex genetic mapping in this paper to allow us to determine where on this locus the shaker gene is located. And to do so, the researchers isolated genomic DNA from the shaker locus, and they use mutations or breaks in the DNA as markers to essentially create a map on this 16F region of the X chromosome. And this process is called a chromosomal walk. And basically what they do is they connect these different fragments, and they're able to find overlapping components and determine approximately where in this locus the gene is located. And what they determined was that this locus is approximately 80 kilobases from the telomere, and 130 kilobases from the centromere. And through techniques such as Southern blot analysis, which runs DNA fragments in a gel and separates them based on size, which these fragments can then be stained and analyzed, and hybridization techniques, which uses antibodies as tags to identify specific parts of a certain fragment of DNA, the researchers were able to isolate the Shaker locus to a pretty exact region on the X chromosome. They then went on to find out that the Shaker transcript is composed of about 65 kilobases, so 65,000 base pairs, that can be alternatively spliced to form different forms of mRNA, therefore making different proteins. So what alternatively spliced means is after introns have been removed and when an mRNA transcript is created, essentially the mRNA that's produced can be selected from a variety of different combinations on that initial DNA transcript, or I guess that just basically exon transcript. And so this provided information to the researchers that the Shaker component of the potassium channel isn't one single piece, but it could be several different shapes of protein. And so because there's alternative splicing, there can be variation in what this protein looks like. And so the researchers supported their hypothesis that the Shaker locus codes for a component of the potassium channel through genetic analysis, and they used some homology analysis to look at other ion channels and their similarities, and they went on to further suggest that DNA transcript can code for several variations of a component on the A channel of the potassium channel. So it's the A component that the Shaker locus is coding for. So lastly, the Schwartz paper takes us sort of full circle here, and it allows us to use information gained in the previous 3 papers to analyze tissue-specific structure of the Shaker channel and where different alternatively spliced transcripts may be more present than others. So this paper, interestingly, gets a little bit more into the neuroscience of where Shaker is located in the brain of the fly and how this subunit is found at different levels in different cell types of brain regions. So what researchers did to see this was they used antibodies to stain different regions of the brain for Shaker channels. And when they observed that there appeared to be different regions in the brain that lit up much more than others, they questioned whether this was a result of simply having more membrane in these regions, therefore it would allow there to be more potassium channels, or whether Shaker was actually just more involved in these regions. And so by applying an anti-HRP stain, which basically just stains all membranes of neurons in the brain, they saw that, for example, in the medulla, anti-HRP stained the medulla evenly, whereas Shaker antibodies did not, suggesting that Shaker is just more involved in certain regions over others. Through this analysis, the researchers were able to prove that Shaker gene encodes a component of the potassium channel, and interestingly, that the potassium channel components are much smaller than cloned sodium and calcium channels, which was unexpected as we assumed they would be sort of all of similar size. Additionally, their analysis reveals that the distribution of potassium channels encoded by Shaker is not uniform within the nervous system, but it's actually more heavily dispersed in certain regions over others. So overall, it's pretty interesting and kind of fun to be able to follow a sequence of papers in a historical timeline as we've been able to do here. So I've spent a good amount of time trying to dig through these, and there's a ton I still don't understand and a lot of genetics that still really confuses me, and I'm sure you guys will come across a lot of stuff that confuses you as well. But it's good to get into it and try to figure out what the papers are saying and what the figures are going over, and to learn some techniques that these researchers use. Something that might be applicable to get you interested in reading these papers, or at least to maybe use them as practice, is the importance of this type of reading comprehension for exams such as the MCAT. So after taking the MCAT this past summer, I would say the number one most important skill to have going into the test is undoubtedly the ability to understand and read scientific literature and then apply with what you've just read to questions. And so it's not pointless to read these papers, and it actually is really good practice for not only this class but for the future. So I hope that helped a little bit. If not, sorry. But yeah, have a good weekend, and send me or Dr. G an email if you have any questions about either of these papers.